Good morning, good afternoon, good night, whatever. Good day, I guess. Um, so welcome to this discussion on sex as a biological covariate in genetic studies. So I've organized this presentation as a Jupyter notebook. So what I'm going to do is I will just go through the Jupyter notebook on the screen, and what you will see is something that is in between coding session in R and PowerPoint presentation, something of the equivalent. And after this presentation, we will put this Jupyter notebook onto the website so that you can tweak it or twiddle with it and change things as you see fit. So just as I know there are a lot of people here, and many of them are actually experts in some of the things that I'm going to talk about, maybe. So please, please chime in as you see fit to make a comment. If you're not an expert, if you want to ask a question, please feel free to do so as well. I just noticed Carl Broman just joined the conference, and I think he's an expert on many of the things that I'm going to talk about here. So here's the plan for what we are going to do today. I'm going to take an overview of Qtl studies, what the underlying principle is. This is both a review of what we covered initially in a previous lecture, and also a preview of where we're going to go in the future, in a future lecture in this series. And in particular, I will, do a quick overview of what the, what is a Qtl study? How are we thinking of it in terms of causality? I will introduce some causal diagrams, and I think those will be very useful in thinking about the problem, and also will be a preview of what we are going to talk about in greater detail in future lectures. So I'll probably get a little bit ahead of myself, but we'll see. Yeah. The things to consider for us are, what are the design considerations and what are the analysis considerations when we are thinking about sex as a biological covariate in Qtl studies. And in particular, we will look closely at the structure of the X chromosome, because the X chromosome is what determines sex, and then examine its implications for the proper analysis of the X chromosome. So that's kind of the rough overview of what's in store. So why is sex as a biological covariate important? It's important for several reasons. For kind of purely pragmatic reasons, NIH has this line as the explicit criteria.
Saunak, can I interrupt just for one
yes.
second? I think your microphone is actually causing more problems. We could hear you fine without it.
You cannot hear me? Okay.
Yeah.
Oh, okay. Is this better?
Yes, everyone will just have to turn up a little bit on the volume.
So to recap, why is this question important? The first reason is that NIH has an explicit criteria, sex as a biological covariate. So if you are submitting a grant, NIH will ask you to address it. And typically, most reviewers are just looking at a sex balance and the implications of that in the study. For genetic studies, there are some additional considerations because of the nature of the X chromosome. the fact that the X chromosome structure depends on sex, and sex is also the phenotype that's caused by the X chromosome. So that's why we need to think about a little more carefully about the role of sex. So first, just a brief recap of what the idea of Qtl analysis. My apologies for the window disappeared from my screen. So a brief overview of what Qtl studies or experimental crosses are and what they mean for us. The basic idea of making an experimental cross for a genetic study in animals is that if you have, say, two rat strains, two strains of an organism that show consistent difference in a phenotype in the same environment, then you know, or you're pretty confident that the difference must be genetic, because that's the only difference between the two between the two animals is the strain, or is the genetics. The environment is the same, and therefore you can say that the genetic structure must be the cause of the difference in the phenotype. So when you cross those two, say, rat strains, then you will create a population of approximately randomly shuffled genomes, and this is due to meiosis, and we are saying approximately random because that's one way to think about how gametes are formed from the parents. Of course, this thing is not exactly random, but that's a separate topic. But for our purposes here, we will consider it as a population of approximately randomly shuffled genome. And because of this idea, any association between phenotype and genotype, you can interpret that to be causal, because it's like you're doing a randomized experiment where you're randomly assigning genotypes to individuals, and then you're looking at the phenotype. Therefore, it's like a randomized study, randomized trial of genotype, and therefore you can infer causality. So examples include backcross, intercross, heterogeneous stocks, recombinant inbred lines, collaborative cross, VXDs. These are all examples of these experimental crosses where the genomes can be considered approximately shuffled. And the definition of Qtl is our quantitative trait loci. These are genetic loci that have been identified to be contributing to variation in quantitative traits. In practice, these traits don't have to be quantitative, strictly speaking, because you can treat a qualitative trait as a quantitative trait. So, in the end, as a shorthand, we're going to say Qtl. So here is a schematic taken from the ratgenes.org website. And you can see that here's an F2, there's strain 1, strain B, strain A, strain B. You make an F1, has one chromosome from each strain. These animals are heterozygous, but still genetically identical. When you make the F1s to themselves, then you have a population of approximately shuffled genomes. Note that nearby loci are correlated. That's because of the way genomes are being shuffled here. And on the right here is a schematic of how heterogeneous stock population might be derived. Here you have 8 parental strains, and there is pseudo-random mating for several generations, and you end up, each animal is a mosaic of these 8 strains, still approximately randomly shuffled genomes. So now let's just look at the structure of the problem, the causal structure of the problem. And here I'm going to use what are called directed acyclic graphs. And this is going to be something that we're going to talk more in a future lecture. But for the moment, I hope these arrows are clear. Are these arrows visible to you? Yes, no?
Yes.
Yes, okay. So here's the causal structure of Qtl on an autosome. So what you can observe is the phenotype and the marker genotype on an autosome. And what is hidden from your view is the Qtl. You don't directly observe it. And what you know is that the process of meiosis on that chromosome is what caused the state of both the Qtl, the locus that is contributing to the trait, as well as the marker on the same chromosome. That's the common cause for both of those. So in this case, if we cannot directly observe the meiosis if you don't know that state, then if you're only observing the marker, then, and even if the marker is not directly causing the phenotype, because of this link through the common meiosis of the Qtl as well as the marker will show an apparent association with the phenotype. Okay. If you could directly observe the Qtl, then it's just like a randomized trial, and then you can directly observe the association between Qtl and phenotype. So in the Genome Scan, what we are doing in Qtl Studies is we are taking basically all markers, or very closely spaced markers on all along the genome, and markers that are on the same chromosome that is share the meiosis with potential Qtl will show an association. That's the basic idea behind Qtl analysis. So for the, I will kind of suppress I will consider Qtl and marker to be almost synonymous in some of the future slides, but with the understanding that really it is close to a Qtl, or they share a common meiosis, but they're not necessarily the same. Okay, now let's think about sex as a biological covariate. So there are some philosophical questions, or maybe not completely philosophical, but at least some conceptual questions. Is it a covariate, that is an independent, manipulable cause? Or is it a phenotype or an outcome? Or is it a mediator? Or is it basically a vehicle through which a phenotype is being caused? So the answer to these questions would depend on the design of the study and the nature of the phenotype you are studying. And so let's think about this a little bit more. So now let's look at what might be this causal diagram if we think of sex as a covariate or as an independent cause. So here, this is a diagram of how it might be. So here I now have two meioses because I'm assuming the Qtl is on an autosome and the meiosis on different chromosomes are independent. So here, the meiosis on the X chromosome is what's causing the sex, which could cause the phenotype. And the meiosis on the autosome is causing the Qtl, which also causes the phenotype in this case. So in this case, in this way of conceptualizing the trait, you would say that sex is an independent cause. And so here, notice that there is no direct connect. One should not see an association between Qtl and sex here. Because the association, because the causes for the Qtl and sex are independent here. So this is, and of course, you know, depending on your design, you may condition on sex. That is, you might have different numbers, or you may design the study so that you have a certain number of males and females, in which case you're, or just males or just females, that would be equivalent to conditioning on sex. Okay, so in this case, not adjusting for sex will not necessarily lead to bias, but it may be a less powerful approach because you're not eliminating an independent cause of the phenotype, and therefore your kind of error term is bigger than it would normally be. Okay, so now let's look at, this is a more complex scenario where I put in the marker just to emphasize that we don't directly see the Qtl, but we are really seeing the marker, but I think we can neglect that for a second. Now let's look at the case when we do have Qtl on the X chromosome. So in this case, the causal diagram will look like this. The meiosis on the X chromosome is now responsible for both sex as well as the Qtl under that you are looking at, or the genetic locus you're looking at. This common cause is now going to lead to if you don't adjust for sex here, then, and if sex contributes to the phenotype independently, then you're going to see, or you have the risk of seeing an apparent Qtl when there is none because of this route to the association. So this is one way of thinking about why for X chromosome Qtl, we need to think about the role of sex a little more carefully than we would do for autosomal Qtl. And in fact, the kind of a tragedy is that the large fraction of studies completely ignore the X chromosome because of, you know, just, it's a hassle, let's forget about it. I think there was an article, I think by Terry Maniolo, if I remember correctly, in the context of, you know, diversity in genetic studies, and she was writing that, okay, you know, we don't have to solve all the problems. Let's start with something easy, which is let's analyze the X chromosome correctly. So, so this is one way of thinking about why it is important to think carefully about where does sex fit in the study design, where does it fit in the structure of the trait, or how you think about the structure of the trait. You may have noticed some R code already. Now I'm gonna import the RQTL package by Carl Broman, who's in the audience, and we're going to see a few more things. So I'm going to analyze some data as an example by Leah Soberg and colleagues who made an F2 cross between the F344 and WKY rats. This is the example that we used in the previous introductory Qtl lecture, so it might be familiar to some of you. The idea was that the V-star rat exhibits physiological and behavioral similarities to endophenotypes of human depression. And so they made a cross between these two strains of rats and did a Qtl F2 cross, and to examine the genetic connections between behavior and physiology. And here is the paper, one of the papers that was based on this. So I just import the data, and you can see that there were about almost 500 individuals, 112 markers, 25 phenotypes, and these are the phenotypes. In particular, I want to draw attention to two traits here, sex and paternal grandmother, PGM, and which are roughly equally balanced here. And more on what paternal grandmother is in a little bit. But for the moment, just notice that it's roughly equally balanced between the two sexes. So here is, and I'm going to just focus on glucose. It's the trait measured after a challenge, because the glucose levels are thought to be a response in the after the challenge or the stress, because that's a response to the stress. So here is a violin plot of the of glucose, log2 glucose by sex, and you can clearly here males are coded as 1, females as 0. So here there's very obvious relationship between sex and the trait. In particular, the males have a higher glucose level. You can also make a plot by glucose and paternal grandmother. Here, if the paternal grandmother was WKI, KY, it is coded as 1, otherwise it's 0. And you can see that in this case, the difference is mainly between the sexes, the first digit 0 and 1, and not so much in the paternal grandmother. But just to look at it a little more carefully, I just did linear regression using sex and paternal grandmother as interacting, and the interaction term is as a p-value that's 0.2, and so for the moment I'm just going to focus on sex. So what is now we're going to do a genome scan. So what is the genome scan? So what we're doing in a genome scan is you have a null model, which is that the phenotype is just noise, versus an alternative at any given locus that the phenotype is the sum of a Qtl effect and an error term. And we compared the alternative model to the null model at every candidate location for the Qtl and make a test. So we can do that in this case. Let me run this code again. Okay, next I run this genome scan and I run the permutations to get the statistical significance. And if I do that, I get here's the genome scan that I get in the end. So you can see it peaking on chromosome 1, maybe a little bit on 20 and 5. And then I run permutations. In about 1,000 permutations, the 5% threshold is about 3.6, 3.7, and the 10% threshold is about 3.3. So these are the two thresholds which you can see. So basically, maybe there is between 1 to 3 Qtl here. So now let's look at sex as an additive covariate. So here we are thinking of this scenario. We are thinking of this scenario. So here adjustment is really to enhance the signal, not so much to reduce bias. So to do that, in Qtl, it is very straightforward. You just have to specify that there is an additive covariate called sex, and the null model is saying that the phenotype can depend on sex plus an error, and under the alternative, when there is a Qtl, the phenotype depends on Qtl as well as sex in an additive manner. And so what you do in Qtl, you scan for the trait, you specify the phenotype column, and you just state that the additive covariate should be sex. And if you do the permutations, and you will see that this is what the new figure looks like. Okay. So the blue one is with sex as an additive covariate, and the black one is the previous one. And what you can see is that for the most part, the blue figures are a little bit higher. There are some exceptions, but there are few. For the most part, the blue curves are a little bit higher than the black curves. This is consistent with what I said earlier, that it's not so much a question of bias, but enhancing the signal when you adjust for sex here, for the autosomes. The log thresholds are not very different. And in fact, I should probably plot let's plot the thresholds. Okay, so you can see that now, previously I had between 1 to 3 Qtl. If I look at this here, there's one that clearly exceeds 0.05, and there are two that are at or near the 10% threshold. Now, after this adjustment, I have 3 Qtl that are above the 5% threshold. So that's the advantage of adjusting for sex and as an additive covariate when you know that it has a strong independent effect. Okay, I want to go back here again, here, this causal diagram. Now, in this diagram here, there is nothing, does not specify the nature of the effect of sex and Qtl on the phenotype. So as we will see later, you know, the additive model that I just used is an assumption, but in particular, the model that specifies how Qtl and sex can contribute to the phenotype can be more complicated. It doesn't have to be an additive model, like I just said. So the next thing we can do is we can consider a more complex model for sex, which in this case would say, let's consider the possibility that sex modifies the effect of the Qtl, or there is the effect of the Qtl depends on sex. And so this would be called, considering sex as an interactive covariate. So at this point, it is worthwhile thinking about what really we want. Do we want to search for deviations from additive effects of sex and Qtl? Or do we want to find Qtl allowing for a more complicated model for sex and Qtl, as I was saying? The causal model that we had up there doesn't necessarily say that sex and covariate sex and Qtl are additive. So there would be a split in your analytic strategy depending on how you want to think about this problem. So let's consider the second one. I find this to be a little more, well, I like this a little bit better, but there is a price to fitting this model because it is more complex. So here, what you would do is you would run the genome scan, and in RQTL, you would just specify that there is an interactive covariate called sex. And in this case, you would permute the phenotypes within each sex, just as we did last time. And if you do that, then you can see that your log thresholds are now a little bit higher. Previously, they were about 3.6 or so. Now, they are almost 1 log threshold higher, 1 log unit higher. And if I make a plot, this is what it comes out to be. So now we are back actually to the previous scenario where there is one clear Qtl and there is maybe a second Qtl that touches, and these are actually even smaller now, the 10% threshold. So there. If you want to search the genome with a more complex genetic architecture in mind, then the bar for declaring a Qtl is a little bit higher. And so this strategy actually is less successful than assuming an additive model for the Qtl. And Qtl and sex. If you want to do the other strategy that I wanted to mention earlier, that is you are not searching for Qtl with a more complex sex Qtl interaction model, but just want to search for deviations from additivity, there's a slight trick here that you have to do in our Qtl. Basically, you fit two models, one with an interactive Qtl and one with additive Qtl, and you take their difference. And that would be the test for additivity of the Qtl. And there's you have to set the seed before each of the permutations, and what that does is it makes the difference in the permutations meaningful. So it's just and so if you do that, then you will, this is the Qtl searching for deviations from additivity and the corresponding permutation thresholds. You will see that there are two places where you are confident that there is some deviations from additivity, which is chromosome 8 and chromosome 10 here. Now, in a way, so if you could have done two genome scans, one that is adjusting for sex as additive covariate, another is a test for additivity. If you're basing your conclusions on these two, then you have two, you're making two independent tests. So your nominal false positive rate would be a little bit higher than your actual false positive rate would be a little higher than, let's say, 5% if you use a 5% threshold for both these tests. So now let's look at the other case. So all of this is good for autosomes, but what about the X chromosome? So for X chromosomes, what we have to do is we have to look a little more carefully at the structure of the X chromosome, evaluate the consequences, and then look at the examples. So this is now a figure from a paper by Carl, who kindly figured it out for all of us. So let me make this slightly smaller. Okay. So this is the structure of the X chromosome when you make all the four possible F2 crosses that you could make between strains A and B. So let me explain what these are. You may have to think of, meditate over this diagram perhaps later. So let me explain what this is. So with an F2 cross, you can make an F1 two different ways. The female, which comes on the left here, one F1 can be A times B. The other F1 can be B times A. So there are actually two kinds of F1s you can make from A and B strains. And because there are two kinds of F1s, there are 2 times 2, 4 different kinds of F2 crosses you could make. So the first one is when you mate A by B to A by B. The second one is B by A to A by B, A by B to B by A, and B by A to B by A. And the structure of the X and Y chromosomes will be a little bit different in each one of those cases. So let's look at it a little more carefully. So let's look at A by B and A by B. So here, this is the this F2 cross, the maternal grandmother is A, the paternal grandmother is also A. The maternal grandfather is B, and the paternal grandfather is also B. So now let's look at this. So when you mate A and the B, the F1s for F1 females just look just like an autosome. They each have an A and B chromosome intact. The F1s have F1 males have a X chromosome from the A string and Y chromosome from the B string. So when you make these two, your F2s will always have an intact intact A chromosome and a recombinant A chromosome, the females, and the F2 males will have a recombinant X chromosome and a Y chromosome from the grandfather here, which is a B. And similarly, you can go through all the others. Now notice, I just, now notice a few things. First is that for the females, all the females have one recombinant chromosome. So this is, in other words, there is the, it does not look like a regular autosome. The second thing is that for females, that there are basically, or it kind of looks like a backcross in some way. You know, the X chromosome has two possible states: a heterozygous and a homozygous A in this case. And similarly for the males, because there is only one recombinant chromosome, and so there is only two states, two like hemizygous A or hemizygous B. So, you know, just right here, you can see that the genetic structure of the markers on the X chromosome will be different from what you would see in a regular autosome. So what is the impact? And there's a similar figure for the backcross, which I will come back to maybe if there are questions or. So here is what are the implications are. So males are hemizygous because they pass on their X chromosomes intact. And the genetic structure of the X depends on both the cross direction and sex. So yeah, that's the other thing I think I should point out. That notice here, look at this versus this. So here, the females have are heterozygous or homozygous A, and here in this cross, the females are heterozygous or homozygous B. So, and what is different between these two is the paternal grandmother, which is here B, and here it is A. So that's why the recording the paternal grandmother is so important. So what are the implications? So let us consider a hypothetical A by B cross, where the trait has a sex difference but no genetic effect on the X chromosome. So in other words, here are the males. The males have genotype hemizygous A and B. I'm like writing AA and double B. This is what a lot of genotyping programs, this is how they might return the genotypes to you. The trait is basically 20 for each of these genotypes, so there is actually no genetic difference. The females have, again, no difference by genotype, but they have a lower phenotype value, 10 and 10. So if you ignore sex and aggregate the phenotype, you will have phenotypes, roughly 15 for AA, 10 for AB, and 20 for BB. In other words, you will see an apparent effect of the genotype at this locus on the QTL, when in fact there isn't any difference when you stratify by sex. Really, the sex is causing the difference in the trait, but you are, by ignoring sex or not stratifying by sex, you are seeing an apparent X on the X chromosome when actually there is none. And a similar thing happens when, for cross direction, a similar type of effect or is observed. So what is the solution? So first thing is that you need to record the cross direction and sex in order to do a proper analysis, because you will need to adjust or stratify for that. And in particular, you will need to rethink the null hypothesis. So basically the idea is that instead of collapsing the two, you will have sex-specific or cross-direction specific hypotheses. So stratifying by males and females in this case. And if you do that, now you have 2 degrees of freedom, one for the males and another for the females. So you have to rethink the null hypothesis by stratifying by sex or direction. And you will have to rethink and because of that, you will have a different degrees of freedom, which can be 1, 2, or even 3 for an F2 intercross if there are all sex and directions available. And the second thing is that because the genetic structure, recombination density, in particular on the X chromosome is different, in particular is half that of the autosomes, you will need to adjust your significance threshold for multiple comparisons for the X chromosome for that reason. So, and for both of these reasons, the way you conduct permutations needs to be reconsidered. You need to stratify and you need to take into account the different structure of the X chromosome. Fortunately, all of this has been figured out for in our QTL, and all you need to do is tweak one simple flag. So in this case, you can do it by, you do the genome scan as usual, and you add a flag, which is X specific permutations equals to true. And if you run it, then here's what you see. The normal permutation thresholds were 3.6 for the 5% threshold. Now there are 2 thresholds, one for the autosomes and one for the X chromosome. And the autosomal threshold is slightly lower, and the X chromosome threshold is a little bit higher. Computationally, it approximately doubles your computational load because you need a lot more permutations on the X chromosome to get the right threshold. So in summary, sex is both a phenotype and an independent, perhaps manipulable cause. And this has both design and analysis implications. The design implication, first of all, is that it's useful to have enough animals of both sexes if you want to study sex effects. And the second is that you need to record the cross and sex direction in the data collection, and perhaps if thinking about the direction, you need to be intentional about making all different kinds of crosses that are possible to study the effects that you wish to study. The analytic consideration is that the causal structure of the trait will influence what type of analytic structure you decide you implement in order to do the genome scan. And because of the structure of the X chromosome, in order to analyze X-linked loci, you need to use proper tools to handle the X chromosome correctly. And here are some references. That's all I had to say for now, and I would be happy to answer any questions. Audience members are welcome to chime in with comments, and we can have a discussion at this point. Thank you.
Saunak, during your lovely presentation, I didn't see any questions. I just, so we'll see whether we get any now.
I cannot hear you, what you're saying.
So there's one from Carl, maybe comment on the analysis of sex in the X chromosome in recombinant inbred lines. And Carl, you can feel free to comment on that yourself.
I think, Carl, maybe you can answer the question yourself.
Let me make sure you can unmute it. Carl, can you unmute yourself, or do I have to do that? Ask to unmute. Okay.
Carl, are you there? Okay, yeah, Carl is unmuted, but we cannot hear you.
Okay. Well, I guess it's up to you, Saunak. Say something.
So I think for recombinant inbred lines, I think it will depend on how the recombinant inbred lines were derived, right? So, for example, let's look at the structure of
And I assume that Saunak's answer will also apply to panels of inbred strains. Perhaps it's, no, maybe not. Maybe this is specific for recombinant inbred.
If we are making recombinant inbred lines by repeatedly mating these individuals, then actually I haven't thought through what would happen with the males would always be from the B, right here, and the females would have a slightly lower recombination density ultimately than compared to the autosomes. So I guess for recombinant inbred lines, that means that the structure of the X chromosome will look very similar to the autosome, except that the recombination density will be different. There will still be two states for the genotypes, the two homozygous genotypes, but they will be, the recombination density will be lower than the autosomes. So, yeah.
Thanks, Carl. I do see you're unmuted, but we cannot hear you. I had a question, Saunak, and maybe this is a too fine a point, but for the pseudo-autosomal region where we have in mouse and human anywhere from 5 to 10 interesting genes, do we have to treat the pseudo-autosomal region in any different way?
no, so your question is about the pseudo-autosomal region?
Right. So at least in the new assembly of the mouse genome, MM11, I think now has about 8 protein coding genes in the pseudo-autosomal region. It's maybe not worth making it even more complex, but I'm just wondering whether there are things we have to worry about there that are unique to PAR.
The pseudo-autosomal region is really terrible. It's even worse than the rest of the X chromosome. I mean, in females, it acts basically the same, and I think there's very little recombination in that region in females, whereas in males, the segregation of the alleles in the pseudo-autosomal region vary by position. You know, if you're at one end, you look like an X chromosome locus, where you're mostly getting, you know, the male is mostly getting the allele from one parent, and if you're at the other end, then it's it's more like, I mean, how frequently you get the grandfather versus grandmother's DNA depends on where you are in that region.
Okay. That's sort of, I can kind of intuit what you're saying. If I ever map
There's a nice paper about the pseudo-autosomal region in human linkage analysis by Jose Dupuy.
Okay.
And it made me want to have mouse and rat geneticists continue to ignore it just because it's so hard.
Carl, do you mind like sending me a link or the reference? When I update the notes, I will add that reference to it.
Sure, I'll look for it. So I had some technical difficulties in the discussion about recombinant inbred lines, but I guess I was wondering about, I mean, I was just inclined to mention treating the male and the female measurements in recombinant inbred lines as kind of like a bivariate trait, or in treating them as taking the average of the two sexes and the difference of the two sexes.
Right. Yes. So the yeah, I think that I would say the latter. That is, I think it's easier to analyze it and split it into two, like the sum and the difference. I think when we were discussing the structure, genetic structure of the X for recombinant inbred lines. So you made a new point. That's good. Yeah, actually, that's I'll add that to the notes as well. Thank you.
So Robert Butler asks everyone, I've already mapped PARs for rat. R-NOR6 has a really rough PAR, but the new rat assembly that's just about to be released or has been released has a much better PAR. I don't know that it's quite as good as human or mouse. The mouse PAR was pretty lousy until about a month ago when the new assembly was released. We still have quite a bit of time. We've set aside up to half an hour for discussion on this topic. I do have some questions, but let me hold off and just let everybody else, because we've got 30 people on the line still who might have questions. And I can see Saunak any day, theoretically. So hearing no questions right away, let me just sneak one in. I found it really interesting, Saunak, that you did the permutation separately for the X chromosome when you were adding it both, I guess, both as an additive and an interaction. Maybe it was more important for the interaction term. To what extent does that logic apply to all autosomes or different parts of autosomes? In other words, the idea of using a uniform permutation result across all autosomes or across the entire X chromosome, given what we just heard about PAR being an even doubly problematic part of X, why don't we compute permutations pointwise across the genome? Is it not necessary, or is it just computational overhead? And
Yeah,
Carl,
it will be if you wanted to, first, I think the reason why it's necessary for the X chromosome is mainly because the density of recombinations is different, radically different for the same nominal genetic length. So that's the main kind of driver of the difference in the threshold. Across the autosomes, that effect is likely to be smaller. The main difference you might observe in the thresholds is because of marker density. Carl, I think you might have additional thoughts here as well.
I think that the main reason that you need to use a separate threshold on the X chromosome is when the degrees of freedom for the test on the X chromosome is different from that of the autosomes. So for example, if you do a backcross with both sexes, the test that you're doing on the X chromosome is going to be 2 degrees of freedom versus the autosomes, it's 1 degree of freedom. Because you're kind of and that, so the LOD scores will tend to be larger on the X chromosome, even in the absence of linkage.
great, thank you.
In the gene network data, has there been any kind of survey of the kind of nature of traits that show biggest sex differences or sex differences in QTL effects?
Not that I am aware of. So that would have been a topic that Alyssa probably addressed in her Big NIDA grant with Dan Goldowitz in about 2010, where they did acquire balanced data sets for males and females for about, maybe a total of 250 traits. So I'd have to look in that paper to see how they handled that. I don't think there was ever a sex interaction term, but they may have fit an additive effect. But I think it'd be well worth coming back to that question, Carl, because the genotypes are substantially better than they were 10 years ago. And I certainly don't remember any adjustment for degrees of freedom in that paper. I'm not a co-author on it, so I may be mistaken, but that would be a great place to start. I'll send the paper to Saunak so that he can add that as an interesting test case for doing that kind of analysis.
For recombinant inbred lines, there shouldn't, I think, be a degrees of freedom difference between autosomes and X chromosome.
Ah, got it.
The density of recombinations is different.
That's true.
Well, do we know, like for the BXDs, are there both types of Y chromosomes in there?
You asked for the BXDs, Saunak?
Yeah.
I don't know necessarily the answer to that, but I know for the rats, for our HXB, BHX, that's why we specifically say HXB, BHX is because there are two different Y chromosomes in there.
Yeah, for the BXD, some of them are technically DXB, but it's a sort of very small proportion.
David, could you repeat, please?
For the BXDs, some of them are technically DXB, so have the wrong Y chromosome, but it's a very small proportion.
A total of 8, as I recall, including one of the original Ben Taylor strains. I think BXD30. Is that right, David?
Not sure off the top of my head. I'd have to check.
The other question is like the mitochondrial effects, and that would come from the maternal line, right? So so in this case, all of these have the grandmother is A. That's another piece that could potentially come into play.
Well, the other thing that's interesting in recombinant inbred lines is that in BXDs, they should be 2/3 B and 1/3 D, and DXB should be the other way around on the X chromosome. Because there are three, because there are 2 B X chromosomes and 1 D X chromosome at the F1 generation.
So I think then it's worthwhile for me to update with discussion on issues for recombinant inbred lines. I think I should do that.
Apologies, Laura, I have to sign off now and go to another meeting. So if you wouldn't mind moderating to the extent we still need moderation.
Yeah, I think we've kind of worn out our questions. If there's no further questions, we can go ahead and let people go and let them know that this presentation will be on YouTube. We'll have links on the OPAR website. And in the next 2 weeks, so 2 weeks from today, we're going to do kind of a intro to weighted gene co-expression network analyses. So we'll go through some of the background and then some of the R code for that. So we hope that some of you will join us again for that.